Okay, thank you very much for introduction. And I'm thankful to the organizers for inviting me to give this talk. In addition, I'm thankful to the Professor Wang's group for developing the excellent database APD. Because due to this APD, we are able to develop the number of softwares because it is freely available to the public. So in any case, if users are interested, this is my website from where they can get our web databases as well as prediction servers. We have nearly 300 total servers and databases, and all are free to the public. So now, in this talk, I will focus on 4 topics. One is trends in therapeutics, because nowadays trend is changing from small molecule based drugs to protein or peptide based therapeutics. So is it a myth or it's a reality? I will talk about it. Then I will talk about NTBP3, which is our software we developed a long time back. Prediction method. So I will talk about the. Then we developed the modified version, NTBP2 and NTBP3, which is the latest version, NTBP3. And finally, I will talk about the resources for designing therapeutic peptides. So I'm not talking only antimicrobial peptide. The major issues are there, how to take a peptide to real market. What are the challenges? Let's say whether it will be stable, because natural peptides are not stable in the blood body fluids. So how to make them workable or to clear the clinical trials, that's a challenge. Okay. So first, in order to understand whether it's a reality or really work is going on in the field of therapeutics. So what we did few years back, we create a database called a database of FDA-approved therapeutic peptide and proteins. These are the peptide and protein which has been already approved and which are already in the market. So this is the number we got in this database. The total entries is this much, but 160 peptide or protein are there, already approved, they are in the market. This is the figure for 2017. Recently, we update this database and the number have been doubled. So it means the peptide and protein-based therapeutics have been already approved. So this is a ground reality. So it's not just the fancy word, it's a real market, it's there. Now, question is how to develop the prediction method. So first time in 2006 around, our group thought that we should develop some antibacterial peptide. We searched the different databases. We found this database APD. And from this APD database, we collected the information, antibacterial peptides. Then we removed the redundancy. So this is a standard procedure. So I'm not going in the detail of the standard procedure. Ultimately, we get the non-redundant antibacterial peptides, and we check on the independent data set. So the major challenge at that time we face actually, because most of the machine learning techniques, they need the fixed length vector. They need the fixed length vector. It means they need the features which is fixed in the number. So what we did, we take the peptides and then we take from internal, let's say 5 residue or 10 residue or 15 residue, and same time we take from C-terminal. Okay. Here, the length of our peptides were between the 15 to 60. We remove the small peptide as well as long peptides. Finally, we applied 3 techniques. One is called support vector machine, another is called artificial neural network, and one is the quantitative matrix. That's basically a statistical technique. So we got the performance nearly 92.58% accuracy. We got it. And this analysis demonstrates that which residues are preferred and which residue are not preferred in antibacterial peptides. As you can see, cysteine is more prominent. Similarly, glycine, this lysine, so few residue, they are mostly, they are dominating by the residue which are responsible for stability. Okay. So this shows that prediction is possible. And one of the aim of our group is not to publish the papers. Our aim is to provide the service to the community. So what we develop a web server where you can submit your peptide and our server will predict whether the given peptide is antibacterial peptide or not antibacterial peptide. But sometimes user have the big protein and user want to know which region of the protein is responsible for the antibacterial activities. So we develop the routine called peptide mapping, which will predict which region of the protein is responsible for antibacterial activity. So this is an example, and this paper is heavily cited over the years, and till today, it's getting good number of citations, 30, 40 citations every year. So that shows the importance. Next challenge was how to develop the improved version, because over the years, number of antibacterial peptides are increasing. Can we utilize that information for the development of new method? So 2010, we developed a method called NTBP2, where our focus was on the type of antibacterial peptides, what class of antibacterial peptides. So there is not much difference, only double the number of the training data set. But recent development, NTBP3, which is still not published, it's still in the review, but we develop a method which take the largest possible data. We have taken the data from APD3, Chem3, number of databases, but most of the cases, we are using the data set from the APD. It's have the region, because the quality of the data is excellent. That's the most important part. The prediction, quality of prediction method also depend on the quality of the data. If you will get the junk, you will get the junk prediction. So APD always provide the excellent annotations. We take the peptides from the APD in our predictions. Okay. And this is the, we developed the 3 model, separate for gram positive, means peptide, which work against the gram positive bacteria, gram negative bacteria, gram variable. Because we analyze it, and in our analysis, it indicates that we need the different type of models for gram positive, gram negative, and gram variable bacteria. So this is the new method from our group, which is published. So this is the overall flow of this method, that how it look like that. So I'm not going in the detail. We have tried all similarity-based techniques as well as motif-based techniques, but they have the poor sensitivity. So ultimately, we use the machine learning techniques, and then we develop it, and it is available to the public. What we have done the different, because we try to understand the need of the user rather than publishing the papers. So one phenomena here is just to predict. You give the set of peptides, it will predict which peptide are antibacterial peptide, which are not antibacterial peptide. Second is design. That's most important, because as an experimental researcher, want to know which residue I should to modify so my peptide become the antibacterial, or it have the more potential to be antibacterial. So this design module generate all possible peptides, and then predict which have the best antibacterial potency. So it helps the users to design the better antibacterial peptides. This protein scan will help you in the protein to identify which region is responsible for the antibacterial peptide. So this way you can do the virtual screening. In addition, motif scan, you can search it in your peptides or proteins, as well as from blast based on the similarity, you can do it. So the most important part is This is antibacterial activity, but we are using the machine learning not only for the antibacterial peptide prediction or antimicrobial peptides. Our aim is how to do the better therapeutics using the machine learning techniques. Okay. So here, what we have done that tumor HPD, that's basically tumor homing peptides. So these peptides are useful to deliver the peptide on the right place. So drug delivery, this is important. Another class of peptides is cell-penetrating peptides. Means whether it will penetrate the cell or not, because nowadays peptides are used to deliver the drugs. Okay. Similarly, we have the B3 peptide for blood-brain barrier. And this is important from the toxicity point of view, because sometimes it may create the allergy. So it has to be avoided. We have to check in advance whether my peptide is allergic or not allergic. In addition, there is a different type of toxicity, maybe hemotoxic. Another development we have brought in the market, because natural peptide cannot go into the real life market, because in body fluid, you have the large number of peptides, and natural peptide have the limited half-life. So how you will increase the half-life? You have to do the chemical modification. It may be N-terminal modification, it may be C-terminal modification, it may be L2D, or maybe some other modification. But question is, after doing the modification, can you predict the therapeutic property of the peptides? That's a challenge. So we have incorporated nowadays to handle the chemically modified peptides. So it will predict the hemolytic potency of chemically modified peptide. Similarly, we have the method for antimicrobial peptide, which have been chemically modified. So the trend is going how to make them more feasible to the real life, actually. So similarly, we have the different methods. This method, toxin pred, have been heavily used in last few years, because users want to know the toxicity of their peptide and protein before it fail in the clinical trials. It's better to predict their toxicity in advance, actually. That is your cytotoxicity. I'm talking about cytotoxicity. Another important part, that most of the method, including AlphaFold and other methods, they allow you to predict the structure of the proteins. But none of them allow you to predict the structure of the peptides. And particularly when you have chemically modified the structure of the peptide, what will be the structure of the protein? That's a challenge. So our method, this pep structure mode, will help you to predict the structure of peptide when your peptide is chemically modified. So that's all. Thanks for listening. That's all for today. Yeah, I'm open for the questions.
Thank you so much, Dr. Raghava, for the presentation. Let's go over the questions. Let's start with the audience here. Okay, Monica had the questions.
Good morning, Dr. Raghava. Very nice talk. Thank you. Can you address some of the challenges with the toxin pred and the hemo PI? These seem to have, it's quite difficult to get an accurate prediction. Sometimes LO37 peptide, for example, is not accurately predicted by these and many other toxicity predictors.
Yes, I agree with you that what we predict the potential or high probability that whether the given peptide is toxic or not toxic, because predicting with the 100% accuracy is not practically possible. So what we have demonstrated in our methods, that they are more accurate than any other methods.